So first of all, before we go too far, I promised Ji Zhao that I would show. So Johns Hopkins is looking for a tenure track assistant professor, and Ji Zhao, who was Joshua's advisor, suggested that I might show this to you and say that, you know, some of you might be interested in applying for this position because she thinks the minimal cell is a, you know, it is a big thing, and that you guys maybe should consider applying. So I'm going to, you know, anyway, this is available, and you might write Ji Zhao. I will post this. I will post this on our meeting website, but you'll be able to see this. But for now, I want to get back to what we were in this discussion section. What I would like to talk about are a number of things, and these are just some of the ideas that I've had. I do think it would be helpful for advancement of our organization if we can make the minimal cell a BSL-1 organism. Currently, like all mycoplasmas, not Mesoplasma florum, but all, you know, Mycoplasma genus organisms, it's a BSL-2. But because it is, so Sanjay Vashi made a minimal cell, a less than our minimal cell, a 68 gene knockout that they showed was unable to infect goats. Our organism, we know, is incapable of contaminating mammalian cell cultures. So I will be approaching, I realize I need to talk to the USDA, NIH, and CDC to see if we can get this change made, but I think we have a good chance. There is 2 papers out that support this, and Daniela Bittencourt is working on a 3rd one. So this is something I'm going to try to do, and I will be happy to help any of you in other nations who want to try to promote this. Apparently, it's already done in Germany, James Sands has told us. So this is something that I think would be very useful. Any comments on that from any of you?
Hi, Elibio. Wow, I fully agree. Very useful. That's my comments. I mean, would be very useful.
No, and John, of course, you know all the trouble we had sending these samples to colleagues at Johns Hopkins and elsewhere. You know, they would, you know, and like even with Elizabeth Vila, she's across the street. And we used to make this joke, you had to have a handoff in the parking lot because of rulings at the university till we got it clarified that they didn't have to be worried about it being infectious.
Yeah.
Right. So yeah, anything you can do.
Well, I feel like with the publications, we have a shot at this now. And,
yeah, you. Sorry, John.
This, at least for people in the United States, this would hopefully eliminate the requirement that you have a USDA permit to take the minimal cell. SEN1 will still be BSL-2, but the minimal cell, I think we can make not. And then Daniela showed she knows the genes that you would have to add back to the minimal cell to make it a potential pathogen. But, you know, this is obviously, if you're going to do that, you've got to stay at BSL-2. But for anything else, I think we should be okay. How can we be a more useful, functional, interactive group?
I've got an idea. What should we form a Slack group for minimal cell?
That's certainly doable. I know we have this for the Build-A-Cell organization, although I tend to live on email, but I'm sure that I can work with my IT guys to set up a Slack a Slack channel for Minimal Cell, and we'll see how that goes.
What is that?
So that's like a messaging app that you can use for professional environments. And at least when it regards to my lab or my experience with the single molecule labs at Hopkins, it's been very easy for me to like message other researchers about what protocol they're using or what current things that they're working on so that we can collaborate on similar experiments. It makes it easier to find people and contact them if you're working on something.
So that's an easy thing to do, and I will get that done, and I will email people about this. There's no point in slacking people about it until they know about it. So that's easy. What else could we do that might make us more interactive?
I think that, well, I've got one also. Yeah. Would be nice if we could put some kind of priorities and the steps that we need to follow up and probably prioritize some activities so we in some way we can try to focus in a direction of the whole community to actually put more, I would say, impactant achievements or urgent activities. It might help.
Well, what would you, in your view, what would priorities be, Olivio?
Well, we are talking about membranes. I think my membranes are something really very important. I've seen some images, fantastic. I thought about atomic force microscope of scene 1, scene 3A, scene 3B, in order to contribute for a clue relating function. I mean, just to add what Just Villa mentioned, and several other groups actually are producing some very nice images from the membranes. I just thought about atomic force microscope, which at some stage might help, and I haven't seen much, even if we compare with the native mycoplasma one. Well, just came to my mind. This is one. The other one that I like very much is to use interactive machine learning to actually load the whole genome and try to generate a gene bank that correlate a genome and function, maybe to facilitate further engineering the whole structure or the whole genome, even to mutate. And the results of the mutations ones, which Cas9 would be also very nice to have in a gene bank, and to interact that using machine learning, interactive machine learning, which is probably more functional.
Well, that's certainly something that I don't know how to do. Yi from Synthetic Gestalt talked about this sort of idea a little bit. Yi, do you have any thoughts about that, if you're still there? Okay, well.
There was a presentation early on today.
Yes.
Yeah,
Other priorities that folks in the group think we should consider.
John, I will just make one comment. This is unrelated, but you sharing partially your inbox.
No, identify yourself, please.
Sorry, I am Bogumił Karaś from Western University in Canada. But this was just comment that I'm not sure the intent, but you're still sharing an inbox that is covering part of the
Oh,
slide. So
sorry, I'll get rid of
so
that.
maybe, I'm not sure if you're intending to do that.
That wasn't my intention.
But yeah, I mean, I will have comments later on when it comes to genome transplantation and stuff like that.
Okay. So, my goal in setting up this meeting is that we become more interactive, that we know each other a little better. And as I said, that I'm not the node for everybody's interactions. And I think we should have accomplished that at minimum today and last week. Another issue I want to bring up, so we can do this. I think it would be good if we think about having another meeting like this, maybe in 6 months or sooner. Any thoughts about this?
Hey, John, this is Zan. I accidentally had my mute button on, but back on the topic of measurements, I just wrote down a bunch of things that I know we would really like to have more information about them, and that I've sort of we've gone over to try to look at labeling the proteins and then having like John Hopkins do either super resolution imaging, storm on them. But it would be nice if we had maybe a simpler way to identify, maybe radioactively, label them and just be able to trace them being up, things being taken up by the cell, like in the degradosome, this glycosylation of the enolases involved in that degradosome, but nobody knows. It seems to be different from the enolase that's involved in the metabolic pathway, like that glycolysis, not glycolysis, attaching like a sugar onto it, so it can attach to the membrane to keep it localized. That would be great if we knew anything about that. And I think just in general, how these proteins, the membrane proteins, are distributed around the membrane, because I think if James Sense's people talked, they would have mentioned that the membrane is about half full with membrane proteins, and how they're distributed would be very important to know as well. So any type of an experiment that would give you information about those would be fantastic.
Certainly, most of the people who are at this meeting are experimentalists, except for your lab, Zan. And all of us can be, as we generate papers or data, about the functions of proteins or systems, all of these things, ideally, you would be communicating with Zan's group, either directly or through papers, but ideally directly, so that everyone's findings can be assimilated into your models to make them a more accurate representation of what the cell is.
No, you're totally right, John. And the success that we've enjoyed in some of these models really depends on the integration of experiments, and even and when possible, also some theory into the simulations. It makes them much more powerful. Alone just getting the kinetic parameters was a challenge, but we're just, as I said, lucky that we've had decades of biochemical measurements. Single molecule people are getting into the act on some of the things involving genetic information processing. But yeah, the more information we know, and perhaps maybe setting up a database that's just sort of really geared towards SYN3A or SYN1, SYN3A, that would be extremely helpful, where we would put
that actually exists. And Jörg Jorg Stolke, you missed the talk since you weren't able to, since you were lecturing, but Jorg Stolke's SynWiki database, I think, will be that. And the idea is
Great.
more and more of us will be providing them data so that they can assimilate it into their database, and you can then, take out the data that you can use. But I can promise all of you that, we've had a five-year interaction with Zan's group, the JCVI, and they have been wonderfully interactive, and they're just great collaborators. So I encourage you, as you have data, to reach out to Zan and her team, and they will build this into our computational model of a cell.
Yeah, and John, in that database, besides SYN1, we, as you know, in our paper that we just completed, we always compared the things to things like Mesoplasm florum, and then Bacillus, because you're just trying to keep the umbrella of gram positives. That would be useful to have at least the Mesoplasm florum, since you had several of those speakers here today, and I think on Monday as well, at least when I looked at the list. That would be great to include that.
Zan, I will, mention that in our write-up of the meeting, but, you and I should reach out to Sebastian, and I think he would be happy to work with us on that.
Yeah, which brings up my next favorite topic, the proteomics. When it's done, the field is slowly moving to the single cell. It would be so wonderful to get them, and then do at least 2 or 3 different types of mass spec, because we've noticed on the original data that we had, it was only done with a trypsin digest, which tends to underestimate the membrane protein count. So it would be fantastic to at least do it with the chymotrypsin. I think that's supposed to be somewhat better, but anything that people could think how to get a better feeling for the parts that are really in the membrane, the peripheral membrane components, those look pretty good when we compare them to, a few specific studies, but that would be wonderful.
John.
Livio.
Livio, what about, I know it's the ongoing Cas9 mutations. How feasible will be to intensify the mutations, not only to mutate a gene, whole gene, but part of it, maybe in the 5 strand of 3, maybe in different regions also, in order for which to correlate with function, probably associate initially with the membrane functionality. If we get all the genes and mutate all of them, individually, and then try to correlate with the membrane function, this would probably be interesting.
Certainly, we've shown that we can use CRISPR approaches to mutate the genome when it is in yeast. I don't think we have shown that we can do that yet in the native mycoplasma cell. Yo, can you comment on that, please? Yo Suzuki?
CRISPR genome editing within the SIM3 has not been done. I wonder if homologous recombination is, efficient enough.
I don't think it is.
Right.
And I don't see any of the folks from Bordeaux on the group now. So, or has anybody else thought
Yes,
about
I am here.
approaches?
I'm here, John.
Oh, Pascal, good to see you.
Yeah,
I'm
I'm
trying
here.
to look at
I'm back at
multiple
home.
screens.
Yes. Yes, just about what you just said about the possibility to use CRISPR to make some mutagenesis directly in the mycoplasma. Well, this is something that we are just succeeded that, but not in mycoides,
Pascal,
in another
please.
mycoplasma.
I'm sorry to interrupt. Can you introduce yourself, please?
Oh yes,
Don't know who you are.
sorry. So I'm Pascal Siran from Bordeaux, France, and so we are currently working on different mycoplasma. So we are working on the mycoides cluster. We've been working on this cluster for many years now, but at the moment we are not still playing with SIM3 point. But yes, regarding what you were saying about using CRISPR system directly in mycoplasma, well, I guess this is something that will be possible because at the moment we, as I just said, we succeeded in using some CRISPR system directly in some other species of mycoplasma. So I guess this is something that could be feasible.
Do you see any reason to think that we would have eliminated something in the minimal cell that would make it not work?
No, I don't see. But you know, I think if you want to use CRISPR directly in mycoplasma, you have to import a recombination system.
Which in theory
Otherwise,
is doable.
yeah, this is what we have done. And with, if you want to use directly the CRISPR system, well, you can do it, but it will only cut DNA, so it will not help you so much to make some specific mutants or this kind of things.
Hey, sorry to interrupt. I'm Lorenzo from Wageningen University, and I know that in my laboratory, they actually developed a technology for inducible expression of CRISPR-Cas in bacterial species that do not have inducible promoters, and they show that that helped in actually doing genome editing with CRISPR-Cas in species that are, well, traditionally hard to engineer. It's in preprint, although it's been accepted for publication. I put the link in the chat. Maybe that could come in handy also for mycoplasma. The principle behind it is that by slowing the expression of CRISPR-Cas with this inducible system, you give more time to cells to perform homologous recombination. So probably that could also help in engineering mycoplasma straight in the cell.
Okay. Measurement. Elizabeth Strzalski brought this point up, but thoughts about this? I certainly see that, you know, we should be able to build, you know, if Makoto can make the cells, you know, move and dance, we certainly ought to be able to build into cells sensors that would allow us to monitor metabolism and other things in the minimal cell. But I'm curious, you know, your collective thoughts about this. Elizabeth, Jane?
So John, you mean something beyond like putting in glucose with a C13 at some strategic position in it? I mean that you know, technology seems to be pretty. They know exactly, you know, where if you introduce that C13, what you can anticipate is going to be your product, like for lactate or pyruvate.
I was not thinking so much in those terms, but the idea that multiple groups are using the minimal cell, not so much to study minimal cell biology, but as a platform with that they can build other metabolic processes into it, and then the idea of measurement, if we could also build into the minimal cell biosensors to monitor, to better monitor what's going on when you start potchkeying with the minimal cell.
Mm.
Can I jump in, John, for a sec?
Please do, Elizabeth.
This is Elizabeth Strahalski at NIST. I think I'm hearing that there are three aspects to this measurement issue. There's how do we bring existing measurements to bear on the minimal cell, which I think is what Zan was talking about in part. There's John's point of how do we build measurement, like self-measurement capability into the minimal cell? How do we build an engineer and sensor so that it can measure itself in its environment? And then a third aspect of this is another place that maybe NIST can help, which is what new measurement methods or platforms are needed. And something that's really interesting here is that we have the opportunity not just to engineer the measurement platform, but actually to engineer the thing we're measuring as well, which is the minimal cell. There's so much capability and knowledge here. How do we make a version that not only lets us get at the biological questions we're asking, but also make better measurements to better answer those biological questions? Hopefully that helped and was comprehensible. Thank you, John.
So, you know, the idea, as Zan brought up, it would be really great if we could, you know, here at JCVI, you know, there's a big group that does single cell proteomics and transcriptomics, well, at least transcriptomics of mammalian cells. But could this technology be used to get transcriptome data or proteome data from a tiny, tiny little bacterial cell? I don't know that the technology is there yet, but I could be wrong.
This is James Dobbins, John. We're in the process of doing some C13 labeling experiments for post-translational modifications in genitalium, in pneumoniae, and we could easily include CINT3 in that set of experiments.
Great.
Well, this is really, we're trying to show that the hex oscillation, and there's some other interesting questions in genitalium and as well. But we would include them. Right now, we're planning for 3 different initiating sugars. Glucose, of course. We're utilizing a maltose, which is a glucose disaccharide, and N-acetylglucosamine. And our evidence suggests that the hex oscillation post-translational modification event that occurs on the outside of the cell only occurs when the organism is fed a oligosaccharide. And this is true in, you see this also in Mycoplasma's need for peptides in the media as opposed to single amino acids. They need the peptide or the oligosaccharide to actually import the molecule into the cell for some reason. But that experiment's ongoing, and we'll have results in a month or so. It's really easy to track C13 sugars. They give a very distinctive mass increase in all of our detection techniques, whether it's mass spec or whether it's GCMS, whichever one we use, we can see it. It's really easy to follow. If that proves interesting, we could certainly arrange to track some of the more interesting glycosynthesis or glycodegradation pathways through the minimal cell. In other words. But we'll know in a month or so when we get this set of experiments done. And we will include CINT3. I think we have 3A.
James, I will get you anything you need. The UAB had balked at the original MTA that synthetic genomics required, but nobody has balked at the one now, which just says, you know, if you infect yourself with it, it's your own fault. And if you publish and you can't sell it. But
I've had several MTAs in the past. I think we're working under one for the Genitalium transposon library too, John. So
I'll send this to you again, and I'll make sure that you guys have any cells that you need. But
what is the current strain that everybody wants to use to start with?
right now it's Syn3A.
I believe that's the one we have, John.
Okay, I think you have that one. Syn3B lets you put other genes in easily. So
I would
will
love
arrange
to get my hand on Syn3B if I can. I'm in James's lab to be clear.
John, so take him up on it.
I sent Kevin the MTA a long time ago, and he didn't do anything with it. So I'll send it to you and James and see if you can, or Prescott, and see if you guys can push this through.
We probably can. Like I said, we managed to get the MGen transposon library.
Yeah.
So,
Yeah.
so.
We can do this.
And like I said, we already have 3A, and we're planning the C13 experiments anyway. In fact, has the material come in yet, John?
Sorry, what was that, James?
Excuse me?
Has the material come in yet? The C13 label stuff come in yet?
No, not yet, but it should be coming in soon.
As everybody in the group knows, you know, that certain supply issues have made it difficult, but we will persevere. I would be happy to include you on when we get our data back and let y'all look at it and see what we get from Syn3A.
That would be great. So other measurement thoughts, anybody? I don't see anyone speaking yet.
John, did you discuss with Ramos Dame the story sort of about the nucleoid associated proteins, this dramatic change in the number of HU? It was in the paper that Ramos and I and you were a co-author wrote about folding the DNA in around all those ribosomes that Elizabeth elucidated in her tomograms. But you pointed out that it is dramatically different from a next relative of Syn3A. What was again the test that what would you change? Since I think you had eliminated, when you did the genome reduction, you had eliminated a gene in front of it, I think, and one maybe behind it.
So Fatima gave a really terrific talk. You need to look at this when I can get them all posted. But we are going to make a version of Syn3A that we have restored the gene immediately upstream of the histone, the bacterial histone-like protein gene, and then we'll see what Remus finds. You know, they predict that they will see a lot more longer
supercoiling
interactions in their 3C mapping, and they would like us to possibly alter the expression of some other genes that they think are involved in chromatin structure. And so this is something that, you know, we're going to have to get new funding to do, but this is something that we will be able to do. I got to get me some more postdocs.
Can I add something to this, John?
Please do.
I think you very well summarized what you mentioned before, but what I think is interesting here is also to get to a bit of deeper understanding. So now you want to put back a full gene. I think it would be really nice if we have an understanding of how actually this adjacent gene is regulated. Now, of course, the easiest way is to just put it back, and for sure that will help. And if you get the expression levels up, then one way that it will affect chromosome organization is what Fatima Zahra mentioned via supercoiling. But also, I think you would already predict that if there's more of these HU proteins around, that by making a more compact state simply due to inducing more bends into DNA, you would see like a significant broadening also of these the signals along the diagonal that we have. So also that would already lead to more visible domains. And I think that in addition, sorry, one second. In addition to HU, I think SMC is also really interesting. there's still a lot of it present. I think 200 copies.
Mhm.
But what is a bit
True.
odd is that one of the SCPA proteins that need to bind to SMC is really low. So I don't know whether there's something has also been perturbed, but that may also be something to look into whether that can be restored, because otherwise it might simply be inactive if not both of these units can function together
Right.
with SMC.
Yeah, that's the hinge, Ramos. That'd be very interesting. You make our lives miserable as a theoretician, but because it was hard enough folding your predictions around the ribosomes, but hey, we worked with the truth.
And there's one more thing that I think would be really interesting to add, because I think that John mentioned that there's some transcriptomics being done by Bernard Paulson. And that would be really helpful also, like to get a full understanding of the structure. Because what we realized also when looking at the genome and looking at some other cells, what happens is that RNA polymerases are anyway like considered to be, in some cases, to contribute to structure in different ways, whether it is directly or via their effects on DNA topology. But in E. coli, one thing that is really like standing out there, and that's a fairly recent finding, is that there's a lot of like spurious transcription. So transcription that is just happening because the DNA is AT rich. And what I'm wondering is whether, because this genome is so AT rich, whether this is also not really having a large impact on what's actually happening to the genome, and also like to gene expression and everything that's downstream.
So, Remus, I was part of a paper done by the group in Barcelona a few years ago, where they found that there's a huge amount, because the Mycoplasma mycoides in 1.0 is so AT rich, that there's a great deal of spurious artifactual transcription. We have now done a transcriptome using nanopore long read technology of SYN1, and we're going to do, based on our studies doing that, we're now going to do it with SYN3A. And so we will get that available to the world. And then I will press the Paulson group. They have Illumina sequencing with their transcriptome, but you know, those two kinds of data sets you may find very useful. And then your group and my group and Zan, you know, we should have a meeting to decide exactly what new versions of the genome we need to make for your future experiments.
Yes, well, it's very good to hear that these data are indeed being there or being generated. Yeah, great. Thank you.
So other measurement issues, I'm sorry. Looking at my list here on the screen, future meetings. How long should we wait before we do this again?
Bogumil here from Western again. So I actually, you know, having meeting once or twice a year like this would be great, but I would like to, like, if possible, to maybe even like every 2 months have something almost like a journal club that we present 3 to 5 recent paper, like just 2-hour meeting every 2 months or every 3 months, when people present the newest papers from our group. I don't think so that is too often, you know, even every 2 months for like 2 hours. And that would allow us to, I think, also interact more. So that I would like some of those journal type like thing on our papers, plus like meeting maybe every 6 months, more like what we had today.
So, you know, if you agree with Bogumil, show me a hand. You know, most of you, I can't see you, but you know, you've got the thing at the bottom of the screen that lets you do this. Bogumil, I will see about setting up something like this. It should be easily done. It will require, you know, this group agreeing to do this, but I'm willing to organize the thing. So
Yeah.
that's on my list. And then maybe we'll try to do this larger meeting again. So what, it's the end of September. So we'll try to do this at the end of March.
You know, it might be nice just I know everybody has a slightly different schedule to their universities, but if we could have it like at one of the breaks, or like, our semester ends in May, or, I think this time we will allow a break in March. Before, because of COVID, we just told everybody to work up through spring break and then go home and don't come back, right? But if we could pick a time that's a little bit more convenient, that'd be great.
I will work on that. I realize, there are a whole lot of universities spread around the planet, but I will put some kind of poll out to see how I can make something that works for as many people as possible.
Yeah.
Okay. So starting, today, I'm going to try to write a summary of the meeting, my thoughts, my observations, and a sort of description of our group at large, which I'm hoping to get published in ACS Synthetic Biology, or Jean Picot's Synthetic Biology group. But I will be floating this around to the group and asking for input from all of you. Any thoughts about this before we move on to other topics? It might help expand our organization, which I think would be, more people using this will get us to this idea of, knowing what everything does sooner. Okay. Genome transplantation can only be done with, a few mycoplasmas at this point. And no one has ever, I know Bogomil gave it a good shot with Haemophilus, but we were never, no one's ever had any funding to try to do this with anything else other than mycoplasmas. But it is a bottleneck. So all of you, I think, experimentalists are capable of putting new genes into SYN3B, but almost no one has been able to replicate genome transplantation without learning it either from Carol Lartigue or Naysira Assad Garcia at the JCVI. It is a tricky, difficult technique, and it is possible to put new genes into the minimal cell, but to manipulate the genome, you have to go through a yeast intermediate, and manipulating the yeast isn't that hard, but genome transplantation is a problem. Any thoughts about how we might get around this, or what we can do to make it so either more people learn to do this than the JCVI, or how we might go about this? Right now, the people who can do this are the JCVI, Kazuhito Tabata in Japan, Carol Lartigue's group and in Bordeaux, and Sebastian's group in Canada. I don't think anybody else has made this work. It's just a hard thing to do.
So John Bogomil here again. I think some kind of, I think you mentioned this in the past. We talk about some kind of devices. Could we make some parts of this process automated? So I don't know, either even isolating the DNA from yeast as we prepare the plaque DNA, or few things that could be automated, that people could take that device and at least some of the aspects made it automated. Again, that would be, I think that would solve a problem. I know that the first time I did it, I actually just picked up Carol's paper and I read it, and I get single colony. That was my happiest day in a long time, when I saw that single colony actually came to life just by reading that paper. But yeah, it is very on and off process, and you practice a lot before you actually can transplant. So I think automation of this somehow would make it easier, so people could just take that and they wouldn't have to spend months or weeks trying that. So I don't know if there is anyone is investing any effort into automation, little devices to move those liquids around or genomes around.
Elizabeth, you look like you want to say something.
Bogomil, are you going to start this company to make these devices?
I wanted to do that. This was like one of the very first things when I tried to do it in my little startup. But again, the problem with such a thing is there's very few people actually will pay for it. So the market is extremely limited, right, to us.
The issue, though, is that the yield is so low that it's hard to study it with the measurement platforms that we have. And so it's hard to know what to optimize about it to arrive at that protocol that people can then use in a very straightforward way. Or so that somebody can build the platform to sell it to other people, chip-based or whatever, to get it to work in their laboratories.
I mean, it's just gentle extraction and there's a couple steps, but again, this is not a business idea that we are too small. There's no money to be made there. So it has to be a grant or something that would help us to move forward. But I mean, we know what we learn a couple tricks with the different membranes, with the temperatures. We know we can analyze how good the genomes are, how to harvest. So we know a lot of different things. It's just, I think maybe Elizabeth, with you, just building those chambers, and I think it's going to come to cell concentration in those chambers, like how many E's we have there, because if you have too little DNA, they break. If you have too much, they just like really stack like a glue together. So again, with few people between all of us and knowledge, with John and Carol and your expertise in the devices already, I think there could be something forward, and we could just build at least part of them.
I wonder, Bogomil, is there some kind of master document somewhere, or does this exist in somebody's brain that has all of, has the protocol as it was published, for example, but then has all the kind of tips and tricks, the margin notes, right? Can we make a better, more improved, more robust protocol where everybody's input is there, and then somehow synthesize that into a document where we can all agree this is the best practice? And John, I'm also thinking of, for example, the encapsulation protocol from Build a Cell, for example, which seemed to be very successful. Something similar.
So I am about to send Andras Cook, Andy, on your screen, if you're looking at everybody, we have been unable to replicate it lately in La Jolla. So we're going to send Andy to Rockville to work with Nasira Assad Garcia. And I want to get a video. So Carol Artigue, we made a video of Carol Artigue doing it in 2009, but I think the technique has advanced a little bit, and I'd like to get a new one that is a better made video. Pascal, do you have any thoughts about this? I know your lab uses this technique. Well, probably you don't, since you're not working with mycoides that much.
We're still working with mycoides, but the mycoides cluster.
Mm-hmm.
And so yes, it's definitely, it's not that easy to get efficient in genome transplantation. But I was just listening, and I was wondering, well, I don't know. I don't want to talk for Carol because she couldn't attend the meeting at that time, but I was just wondering if it could be meaningful to organize, a face-to-face meeting coupled maybe with a hands-in workshop. Maybe if there are different groups who are interested in learning the techniques by hands, why not thinking about that?
I think that would be a good idea. Right now on the minimal cell meeting workshop, Kim Wise has done two techniques, videos of two techniques with where sometimes you're seeing Kim, sometimes you're seeing PowerPoint presentations that explains the steps in his techniques. This is for measuring cell growth rates and also for preparing SP4 plates, which there's some tricks to that a lot of people don't know. We have learned this from working with collaborators who work with other bacteria, that they don't realize how temperamental SP4 can be. But I think that transplantation is something that it is not an impossibly difficult thing to do. I think it is automatable. I think in some ways automation might make it work better, because you can make automation systems be more gentle than you can consistently do with a hand pipette. But a video showing the technique with voice overlay and steps in the process would be very helpful to enable people to do this. And also a similar video showing how to use CRISPR now to modify the genome cloned in yeast, as opposed to the TREC technique that the JCVI originally published. Both of those are things that are needed for our community. But
maybe we
broader
can,
sense. Go ahead, Pascal.
No, I yes, I completely agree with you, John. And maybe we should talk this with Carol. So
I will set something up with Carol and get you in this also, and maybe some other folks who would be appropriate. But in a broader sense, so transplantation works for this organism, this minimal cell, but production of other minimal cells for other organisms. So the Church lab, George Church's lab at Harvard, has been trying to make this, E. coli for years, and they are having to do it in a stepwise process of, conjugation and P1 phage, where they replace, 50 kilobases of a genome at a time. And they said they'd love to do genome transplantation if they could. But so if we want to be able to make other minimal cells for other organisms, building the genomes is not that incredibly difficult. It's booting them up. And what is needed, I think, is someone, probably my group, I need to write a proposal for this, but it would be great for microbiology in general and minimal cell studies if we can have a concerted effort, a couple of postdocs doing what Carol Lartigue did for Mycoplasma mycoides, if we can learn to do this for other organisms.
And John, I would just add to this, because there was all this recent paper that are kind of following up the E. coli synthetic genomes with splitting, the fusion and fission of the genomes, when they can have a megabase or megabase, and you can electroporate have a megabase directly into E. coli. So if you can actually split the genome into 4 or 5 chromosomes, or have those landing pads that you're inserting not 50 kb but 500, it is getting actually quite fast process. So this may be always the argument, and again, I think if you're gonna go to other cells that are larger, the handling of the genome, right? Like above, I would assume above like 1.5, maybe 2 megabases, will be huge challenge in addition to all the membranes, right? So, but yeah, I agree. Like if there was any way to get more money to understand the genome transplantation.
If Elibio Reg, if I can comment on that also. Bogomil, I thought about electroporation also. I thought about the gene gun also. The problem is the size. If it's a huge size, it will break when you actually accelerate. Well, we never know, because you should consider that behind the particle per se, all the particles, all the DNA covering the front will break, certainly. But you are talking about billions of molecules, that the molecules inside the part or behind the particle will go through and will be recognized. The other one was about microinjection. I know the cell is very small, will be very difficult to hold, but with a very good microscope, we might be able to do that directly, because to pipette, gently pipette a large size of DNA would be feasible without breaking it. At least you can add that to the cytoplasm, trying to mimic what you remember about the cloning a goat, that you actually get immature embryo, that you remove the DNA and you actually transplant to using a cell with microinjection, and seems to work. You see, at some stage, there are probably quite a few approach that could be tested. What do you think about that?
I personally don't think the microinjections, with those cells, when we talk about those cells, going to be possible, or it's very. I still think that genome transplantation overall works quite nicely with mycoplasma, and a few optimization there. And again, it's just the DNA isolation and how we grow the cell. I know that, for example, we grow them at different temperatures. We move them, I would move them from flask to flask. If we have some kind of incubator that is controlling the cell growth, and then we have some kind of other device that is isolating the plaques, I think that's going to be easier than trying to develop the microinjection. Now, for the giant genomes, like 5 megabases and so on, maybe we can, have the electroporation and electroporating the 500 to maybe 1 megabase or something like that, right? So we could kind of go with that. Particle bombardment, I don't think so. And microinjection for tiny cells, my feeling is no, but that's just my feeling.
Other thoughts about transplantation? Bogomil mentioned that, the cells are too small for microinjection. Elizabeth Strahowski and I have been thinking about it would be ideal if we could make our cells bigger. It would be really great if we could do that. So in the preprint from Jay Lennon and Roy, he talked about it last week. By mutating FTSZ, they were able to make at least SIN1 cells bigger, less so with the minimal cell. But other thoughts about how we might make the cells bigger and more easily imaged, given that we saw from Makoto how possible it is to drop a few genes in and radically change the structure of a cell, what it looks like. So maybe we could put a few genes in and make the cell bigger. Anyone have any thoughts about that?
John, the linear pathway for lipid metabolism in SYN3A, to get either PG or cardiolipids, there does seem to be perhaps a rate limiting step there. I wonder if a mutation in that gene or would help at least make more of the lipids. If the ratio lipid to membrane proteins is critical, that's only half the problem, but that might be just grow more lipids.
So in the Cell paper with SYN3, it looks to me that in the absence of the 7 genes that enable the cell to look like and divide like a normal Mycoplasma mycoides cell, that the cells are producing more lipid, these huge vesicles. So there may be, altering the expression of one or more of those 7 genes might be a key to changing what the cells look like. Although, it's a hypothesis that, we could try, but there may be other ways to do this. James Pelletier was talking earlier today about I mean, is our cell's size determined by biophysical properties as opposed to the lipid and proteins that are being expressed? You know, these are things we don't know.
Could we increase copy numbers of the genome? Because I don't know if there's clear correlation of if we could temporarily increase it from 1 copy to or to like 5, 10. I don't know how difficult would that be. Would the cell become bigger? So at that point we could just inject something or.
Well,
yeah,
the genome doesn't occupy that much of the cell's volume.
But maybe the production of things. I guess that's true.
You know, whereas we have 7 proteins that we think are involved in genome, in cell division, and possibly in control of the membrane accumulation, because we see these huge vesicles made by SYN3, but not by SYN3A.
So what is the volume of the genome that is occupied in the cell?
I should know this, but I don't.
Like less than 10%?
Yes. Actually, Zane Thornburg of my group should be able to give you the exact number, or Benjamin Gilbert. It's it's less than 10. I just don't remember if it's 5 or 8. Are either you guys still there?
I think they're still there.
Yeah. Zane, we can't hear you if you are. You're muted. Yeah, I'd have to look it up in the paper. When we tried to predict the, to fold the DNA around the ribosomes from the tomogram, we were guided by Ramos Dame's lab work on the chromosome capture map. At that time, we did an estimate, how much space was taken by the ribosomes, how much was taken by the DNA. I just don't remember the numbers. I'm sorry.
Okay, that's okay. It's just maybe I also actually am really interested in evolution. What would happen if you have every gene duplicated, and the second gene can take on some different function? So overall, having duplicated genome or more copies would be kind of interesting.
Well, the main problem, we did already in our calculations, we account for when you do duplicate the genome, which seems to happen around anywhere between around average, let's say 60 minutes. And you have a second copy of the gene. There's always a question, which has been looked at only anecdotally in E. coli. Does that second copy of the gene start being transcribed at the same rate as the first one? And at least from the studies of Ido Golding, it seemed like it depended on the growth rate a lot, whether you had comparable transcription rate. But I also will point out this thing, since we really reduced, it doesn't make the amino acids, and you have to also bring in nucleosides or nucleobases to do it. Those transporters have to really start bringing in even more to be able to keep up with the demands of making the deoxynucleotides. And it's not clear it can do that.
Sorry, I might be completely like tangenting off into like another realm here, but one thing that I thought about with the difference between 3.0 and 3A and B is like genetic replication, or rather the I guess like weird cytokinesis and whatnot that's going on. Is I think I saw in a review that M. genitalium doesn't I think it's like up to like 1/3 of like the times it tries to reproduce makes like an incomplete genome set from it. I wasn't sure if anyone's looked at or like tested it in that or like that could be like the model for why those weird reproduction or like those weird, I guess, like little like webs that take place are going on, like incomplete genome content.
What Again, I can only tell you from like some other studies that we did, at least in our model of genetic information processing, whether you duplicate that DNA or not really depends on how successful you are at initiating the DNA replication. That means you have to form this filament of DNA A. And if you run out of DNA A and the cell grows, you will not be able to initiate again. And you could get a longer cell with only one copy, but this is very preliminary. You know, one was, you know, testing out different concepts and because the kinetics of that filament formation, at least what we were using in our model, is from a single molecule experiment. And, you know, they're never done in the cell, they're done, you know, in vitro. So we wondered what would happen. So yeah, maybe if you could somehow inject more DNA A, you can get at least a filament formed, and that would initiate it at least. But you need that to keep the single strand sort of apart so you can get the replosome in.
So I'm just looking at my list in the things I'm in the slide I'm showing. Does anyone have any requests or other things that they want to bring up in the discussion section? Are there things that you need me to do or someone else who might be listening here to do? Now's a great time to mention it.
Okay, I will speak last time. I'm taking too much time today. But you know, if there was, you know, if you're thinking of the next generation, should we put like a course, like undergraduate course? Maybe we could be even teaching in each other's classes or something that we actually, there is just so much development happening with the cell that would allow us to keep summarizing everything and keep adding it. But that's just idea. Could there be, you know, I know there's a number of courses built as circle. Build a course specifically for mycoplasma or the synthetic cell here around it. And this would be leading kind of course that will be upgraded every year. It could be delivered and just recorded, or it could be, you know, anyways, like, you know, since, you know, I have to do some teachings right now that I always think of the students and because there's just so much excitement here, but we are such a small group that we are focusing on this. And then I think the more people in undergrad and grad courses, you know, would be exposed to it, this is gonna even grow better. So that is kind of the side idea. Could there be something for students?
So, you know, I am not in the teaching business except to individuals. Those of you at universities, does this seem reasonable, Bogomil's suggestion?
Oh, this is Zan again. Hate to keep piping up, but oh, definitely. Because I saw the problem that one has, not only just with the students, but the community. For so long, one was just fixated on the results coming in from E. coli. You know, I was thankful that people started to study bacillus, you know, because there are some differences in being gram positive, gram negative, and you learn more and more from these minimal cells. And I think you do have a chance to represent all the cellular networks under one umbrella. That I make this joke that even the physics students in my group can understand how a cell works, right? And I think it would be so helpful. You know, good pictures to go along with it. Hamilton Smith used to say we should write a book on the minimal cell. I think he's right.
Yeah, you know, there's that, but I mean, I'm still getting requests, you know, weekly to write, you know, would I be willing to organize a book that, I mean, you know, about my research. And what I'm wondering, does anyone ever read these books that I have contributed to? Whereas if it was just something that was online that, you know, people could access, that, you know, 10-minute talks or something like we have now. I'm so far out of the education business. I don't know the best way to go about doing something like this.
A 5-minute movie is about what they have the patience for.
Yeah, agree. 100%.
Well, that goes with the minimal part.
No, but 5-minute sections about the different aspects and keep that actually could be perfect. And
Yeah.
could be updated. That I really like that. There could be like, I don't know, 25 minutes video that could be updated, and that would be good for everyone, but also, potentially we could show it in our classes. Yeah.
So if something like that existed, would people use it? Would faculty at universities, include like 5 hours of lecture about minimal cell in a class?
Definitely. I would.
Okay,
Yeah, like
maybe so. Okay, that's useful to know. Anything else? Now's your time. Now's your chance. Let me know. I'll write it down.
I just want to mention, if I can, that videos and undergraduate textbooks are not mutually exclusive and will reach different audiences. So whoever needs tenure can work on, their textbook along with whoever else wants to join in, right? And then we can ask Kate, for example, Kate Adamala, who she worked with for the synthetic cell videos, because I thought that was produced really well.
good idea.
And each textbook can be posted online, available chapter by chapter for download. And that'll be nice too, because that's searchable.
Mhm. Somehow, it just seems like, if we start talking about a minimal cell textbook or book, that I'm likely to be the one who has to do this, which, is not impossible, but it's something I can think about.
Yeah, no, I think
I know I can get help from my friends.
and these short videos from the various groups would help, particularly if we have a sort of a central animation or graphics expert who could help us, right?
That I think I can find in-house at JCVI, and there are folks who would be happy to help me with this and not charge me to do it.
Oh,
So
wow.
I'll look into that a little more.
Okay.
So
John, I wanted to bring up one other thing, and because I have not been able to be here for until the very end, want to really thank you for inviting my students to speak here. They said they learned a lot, and I deeply appreciate your engagement.
San, the minimal cell is my life. I think of myself as a prokaryote.
Oh, my God. Carl Vose, he was my collaborator for many years. No. You're either an archaea or you're a bacteria. You can't be a prokaryote, according to him.